The following podcast is part of a certified educational activity titled Mastering Cannabinoid Science: Key Insights for Informed Clinical Decision-Making. Access the entire activity and complete the post-test at peerview.com/EPE 860. Downloadable slides and practice aids are also available.
Hello there. I'm Kent Vrana, professor of pharmacology at the Penn State College of Medicine and director of the Center for Cannabis and Natural Product Pharmaceutics. I'm delighted to have this opportunity today to talk about the science behind cannabinoids as medicines and to provide some key insights into how we decide to use them and the ways in which we use them. Our goals today are first to introduce you to how the cannabinoids work in the body, in particular that the phytocannabinoids that are found in plants are accessing endogenous systems or the endocannabinoid system in our bodies and how that produces potential pharmacological benefits. I'll then talk a little bit about the evidence and regulatory standards behind that differentiate prescription grade from over-the-counter cannabinoid products, and the fact that this lack of regulation of OTC products or over-the-counter products places our patients at risk. As shown on this slide, there are three main classes of cannabinoids. As shown in the middle panel, what's bringing us here today are the phytocannabinoids. These are molecules derived from the cannabis plant. You're most familiar with delta-9 tetrahydrocannabinol, or THC, or cannabidiol, or CBD, because they've been getting an awful lot of attention as potential medications, as well as the recreational use of THC-dominant cannabis. But what's important to understand is that there is an endocannabinoid system that's part of our bodies. And in particular, what we do is that we use the bioactive fatty acid arachidonic acid, the 20-carbon fatty acid that's found in all of our membranes, and we convert that into two primary molecules, 2-arachidonyl glycerol, 2-AG, or arachidonyl ethanolamine, AEA, also known as anandamide, which, by the way, is a Sanskrit term for bliss, reflecting the fact that the psychoactivity of THC is reflected in its euphorogenia, or its euphorogenic nature. Our bodies use these molecules to modulate our homeostasis. And so what the phytocannabinoids do from outside the body is usurp that system and have an effect for recreational purposes or potentially for medical benefit. And then finally, the third class are the synthetic cannabinoids. Following the discovery of the active ingredients of cannabis and then the active ingredients of the endogenous system, large pharmaceutical companies over the last 40 or 50 years have been trying to develop new synthetic versions that might become more efficacious for treating disorders. And while that hasn't come to pass in general, it has left us with this large library of chemically synthetic molecules that also interact with the endocannabinoid system. These clinically relevant cannabinoids derived from plants, the phytocannabinoids, reflect a very complex system that for the last 5 millennia have been known to have pharmacological effects. And in particular, we know that smoking the flower, the dried flower of the cannabis plant that contains high concentrations of THC, can produce euphoria, for instance. So the flower produces cannabinoids, flavonoids, terpenoids, these small organic molecules that have activities in each of their own rights. Similarly, the leaves, the stems, and the roots have other compounds that might have benefit, but in general, they don't contain high levels of the cannabinoids, THC and CBD. That's generally restricted primarily to the flower. But the idea is that this wealth of chemical diversity that's found in the plant can conspire to create a synergistic effect, also called the entourage effect, where it's not just the cannabinoids, but it's the combination of cannabinoids with other compounds that may be providing therapeutic benefit. We think that there are perhaps 180 different molecules, cannabinoid molecules, that have been identified and grouped into a series of chemical classes. But at their core, what we have is a system in the cannabis plant that condenses geranyl pyrophosphate with olivetolic acid. And that produces the precursor for all the cannabinoids. And it's called cannabigerolic acid. And this molecule serves as the precursor for CBD, the direct precursor of CBDA and THCA. And the A in each of those cases is a small carboxylic acid that's found in the upper right corner of CBGA, for instance. And when these molecules are heated up through smoking or vaping or processing, that carboxylic acid naturally and non-enzymatically leaves the molecule to produce delta 9 THC that we know produces euphoria, and CBD or cannabidiol, which has anti-inflammatory activity and other anti-seizure activities that we'll talk about in a few minutes. In addition, not shown here is a molecule, cannabichromene, that's produced also from CBGA. It has its own activities. But the bottom line is that these three compounds, CBC, CBD, and THC, become a plethora of other cannabinoids that are shown in the lower right side of this slide that are only now being understood for their pharmacological activities. But you will find marketing of CBG, CBN, CBC, these other molecules. But again, we know most about THC and CBD. At the core of this is the endocannabinoid system. That's what we're considering today. This is an endogenous system, the ECS, the endocannabinoid system, that is composed of endocannabinoids, small molecules, and receptors for those molecules. So on the upper right, I just described the fact that there are two enzymes that utilize arachidonic acid. So this is NAPE-PLD that makes arachidinoethanolamine, and Daigle that produces 2-arachidonyl glycerol. These are the two major endocannabinoids in our bodies. Then there are complex and selective enzymes that degrade those molecules. So we make them on demand, 2-AG and AEA, and then we degrade them when we're done using them. And we make them because they interact with selective receptors in the body known as cannabinoid receptors, CB1, the cannabinoid 1 receptor, and CB2, the cannabinoid 2 receptor. CB1 is the most prevalent receptor in the brain. It's primarily restricted to the nervous system, although found in other places, where it is responsible for the euphoria that happens when you smoke a high THC product or you take orally a high THC product. The CB2 receptor is found primarily in the periphery on immune cells and inflammatory cells, where it plays a role in modulating our immune response. Those are the two main players that were discovered in the early 1990s. But there are, in fact, a number of other receptor molecules that bind the endo and phytocannabinoids, the plant-based cannabinoids. And these include an orphan receptor called GPR55. This is a G protein-coupled receptor, as well as the TRPV1 channel, which is the capsaicin receptor, the receptor that gives us a sense of heat in hot peppers, as well as the PPAR alpha and gamma receptors that are involved in glucose metabolism. So in total, these conspire to then provide us with a very rich therapeutic target base because there are perhaps a dozen different receptors that bind cannabinoids. And so for that reason, the ECS is a very attractive target for developing these therapeutics. What's unusual about the endocannabinoid system is that it's retrograde signaling. So what's shown on the left in this slide is a typical excitatory neuron, that blue neuron in the upper left. It releases glutamate, passes across the synapse and stimulates, because it's excitatory, the postsynaptic cell. This is an excitatory pathway. What the endocannabinoids do, represented here by ECBs, they're produced on demand by the target and then go backwards and inhibit the first neuron, the excitatory neuron. And they do that because both the CB1 and the CB2 receptors are 7-transmembrane GPCR coupled receptors that are coupled to the inhibitory G protein. So they suppress activity of the target. In this case, they're suppressing the excitatory pathway. Well, why that's relevant is that recently we have approved by the FDA a purified CBD drug that is used to treat seizure disorders in young children. So how does it work? Well, the thought is that in the normal non-epileptic brain, the endocannabinoid system is predominantly suppressing the excitatory neurons, the ones that might be overstimulating the activity and could potentially produce seizures. So the endocannabinoid system is suppressing that, while it has less of an activity on the natural suppressing neurons, the gamma aminobutyric acid neurons that serve to squelch activity or keep it under control. That's in the normal brain. So the ECS system is essentially acting as a brake. But in the seizure prone disordered brain, what's going on is we have observed that the enzyme that makes 2AG, one of these endocannabinoid suppressing molecules, is decreased. There's less of that enzyme to make the ECB, the endocannabinoid. At the same time, the number of CB1 receptors on that excitatory neuron are decreased. So there's less of a break. And the thought is that perhaps what might be happening is that the cannabinoids can be used to reset that imbalance in the epileptic or seizure prone brain. The efficacy has been proven in clinical trials that led to the FDA approval of CBD as a treatment for pediatric seizure disorders. Well, the basis for this entire story is the interaction of the cannabinoids with their receptors. And so briefly, I'll just point out that what CBD does is it inhibits CB1 and CB2. It inhibits CB1 by serving as a negative allosteric modulator. It does not bind in the active site of CB1, it binds in another site, and it reduces the activity of that receptor. Simultaneously, it is a competitive antagonist or an inverse agonist at the CB2 receptor. On the other hand, delta 9 THC is an agonist. It binds to the receptors and activates them, and it does so at the CB1 receptor and the CB2 receptor. And in both cases, remember that those receptors are inhibitory in nature. So when they're activated, they inhibit their target cells. In a similar fashion, there are a variety of highly selective and very potent synthetic cannabinoids that have been developed as potential therapeutics. And while theirs have never panned out in terms of making it to the market, they are being used illegally or illicitly as super agonists to produce euphoria in the recreational market. The cannabinoids modulate physiological effects through their receptor interactions. Delta 9 THC is an agonist, an activator of the CB1 and CB2 receptors. It has obviously psychoactive effects. THC is responsible for the euphoria in recreationally used cannabis. It's been approved by a variety of regulatory agencies, including in the European Union and in the US FDA, for treating appetite stimulation in HIV/AIDS, for instance, or cancer-induced or cancer chemotherapy, and it appears to have great promise in chronic and neuropathic pain, although it hasn't yet been approved for that purpose. CBD, on the other hand, has been approved for treatment of seizure disorders in children and has great potential for anxiety, post-traumatic stress disorder, and neurodegenerative diseases. And then finally, these synthetic cannabinoids, while used having high psychoactivity in some cases and being used illicitly on the street, they're referred to as compounds like spice and K2, but they also have the potential as anti-cancer agents and in cancer-induced nausea and vomiting. Well, whenever we talk about drugs used exogenously, we have to consider ADME, absorption, distribution, metabolism, and elimination. This is pharmacokinetics. We just finished talking about pharmacodynamics, which is what does the drug do to the body. In this case, we're talking about what does the body do to the drug. And in general, it's important to recognize that there is a considerable knowledge of how the cannabinoids are handled by the body, and it turns out that they're metabolized primarily by three main cytochrome P450s in the liver: CYP3A4, 2C19, and 2C9. And what's interesting about this is that those three enzymes are responsible for metabolism of about 60% of our prescription drugs. So you can well imagine that what's going to happen is that they potentially can interfere with the metabolism of prescription drugs. So as we think about absorption, typically during absorption of an orally taken compound, the first thing that happens is it goes through the liver and is subject to first pass metabolism. For the cannabinoids, that number is very high. Less than 5 or 6% of an orally administered cannabinoid makes it through the liver into the circulation, which is responsible for the reason that most abused cannabis is done so via vaping or smoking so that it bypasses the liver. Once in the body, it distributes because it's a lipid-like compound throughout the body and has a volume of distribution that's remarkably high because it hides out in adipose tissue. This is why metabolites of the cannabinoids will hang around the system for weeks after your last use, and you can fail a drug test perhaps 30 days after your last use if you were a heavy user, because the metabolites hang out in the fat tissue. The metabolites themselves, in both cases, the first metabolic product is an 11-hydroxy compound in the case of THC, or 7-hydroxy for CBD. Those are both active. They remain active. In fact, 11-hydroxy THC is more potent than delta-9 THC itself. But then they are metabolized to carboxylic acid derivatives that then get modified for secretion in the urine. And so the compounds are very slowly released with half-lives on the order of days, which is why drugs can hang out in the system for a very long period of time. It is very important to recognize that cannabinoids have adverse effects that require very thoughtful dosing. CBD in general has a very positive, very beneficial therapeutic index. It takes a relatively low level to have a therapeutic benefit, but it takes extraordinary levels to have toxic effects. And that's why we can give such very high concentration to pediatric patients. But at high enough concentrations following chronic use, we can get some serious side effects, including elevated liver enzymes. On the other hand, THC can be the source of a lot of psychoactivity and a lot of psychological side effects, including dizziness, drowsiness, and dry mouth to start with, but uncontrolled vomiting and negative psychotic symptoms in extreme cases. Now, this is not true of all patients, but it does appear to vary depending potentially on the genetic makeup of the patient themselves. So THC at high doses, especially when delivered via smoking or vaping, can have dramatic side effects. So the delta 9 THC in synthetic cannabinoids may have a very narrow therapeutic index, where only a slight increase in the relative concentration goes from being beneficial to being harmful. This is why it's vitally important that in prescribing these drugs, we start low and go slow and stay as low as possible while having a beneficial effect for two main reasons. In the case of THC, because it might have bad side effects, but in the case of CBD and THC, because they may be interfering with known prescription drugs. And this is very common for a number of different agents, prescription agents like warfarin or Coumadin, or some of the anti-seizure medications uh, like carbamazepine. So we have to be careful. And the simple rule is to start low and slowly escalate until you get a benefit and then hang out there. What we're only now beginning to learn is that individual patient variability in their genetics can affect pharmacokinetics and patient outcomes. So, especially in naive patients, the use of high dose THC can be very detrimental because it has untoward psychological activities. But at the same time, we have genetic variants in our cytochrome P450 pathways that can produce a patient who is at risk for too high a concentration because they're unable to metabolize the cannabinoid, the phytocannabinoid or the exogenous cannabinoid that's being administered. So it just reinforces our notion that we have to go low and go slow when we're first starting our patients. Well, I'd like to switch gears and now talk about the fact that prescription grade cannabinoids, and there are several of them, are held to a much higher standard than over the counter products. And in the drug discovery process, we generally start with understanding in animal models or cell culture, or even using artificial intelligence, what are the potential benefits of these molecules that bind to receptors. But then as we proceed through the drug discovery process, we start to engage patients to look at or subjects, normal subjects, to assess safety, then efficacy, and then the gold standard is a double-blind, placebo-controlled trial to see the efficacy of the drug for a given indication. For prescription grade cannabinoids, this is very carefully monitored. Whereas for the over-the-counter cannabinoids, they are not subject to this kind of control. And so we can have market availability without the careful clinical trial data that I've just described. This is a simple diagram that describes that process. So we get an idea that one of the cannabinoids might be beneficial for a disorder like chronic pain. Well, in order to get it into patients, we have to file an investigational new drug application with the FDA. Then, in phase 1, in a handful of healthy patients, we do dose escalation to find out that it's safe and it doesn't produce side effects. And in fact, we escalate the dose to a point where we first start to see the first potential adverse effects. That is our maximum tolerated dose. Then in phase 2, we bring in patients and ask, is it going to be efficacious for the disorder that we're going to treat? And then in phase 3, the there are thousands of patients engaged in a double-blind placebo-controlled trial where some get the drug, some do not, and neither the patient nor the administering doctor know which one they're getting. And that gives us our best evidence. And in the face of that evidence, the company can apply for a new drug application. And here in the United States, the FDA evaluates that and it approves or doesn't approve that process. If they approve it, it goes into patients, and there's post-marketing surveillance or phase 4, where we monitor that once it gets into tens of thousands of patients, are there any side effects uncovered? This process is required of all prescription drugs, including prescription cannabinoids. And we have a number of prescription cannabinoids, including synthetic THC and analogs of THC that have gone through this process and have been approved to treat nausea and vomiting and to stimulate appetite in cancer patients and HIV/AIDS patients. And then CBD has gone through this process, CBD that has been purified from cannabis and has been tested and proven to be efficacious for the treatment of seizures in pediatric patients. This is not the case for over-the-counter products. They have not gone through this process. So in addition, especially for cannabinoids and THC, they undergo additional testing for abuse potential. Because remember, there is a cannabis use disorder diagnosis in the most recent psychiatry manual, the DSM-5. So there's testing of the interaction of cannabinoids with CNS receptors and neurons, animal testing to look for risk of dependence, physical dependence, and rewarding properties, and then even human clinical trials that monitor adverse effects, including abuse potential, as well as the reinforcing nature of the compounds in using patients, in actual drug abusing patients. Again, once again, this is not done in the over-the-counter products. The source of this problem is, in fact, the 2018 Farm Bill, or the Agriculture Improvement Act of 2018. At the time, the Department of Agriculture wanted to render hemp legal. Hemp is low THC cannabis that is great as a source of feed for farm animals, production of fibers for rope and cloth, as well as other indications. And in order to understand the problem, let me just go into a little bit of genetics. We are familiar with cannabis sativa sativa. This is a plant that makes cannabinoids, but it comes in multiple flavors. And think of the petunia. The petunia is a plant that makes flowers. Some of them are white, some of them are red. It's still a petunia, but it's making a different flower. In this case, the plants are all cannabis sativa sativa. The difference is that if it makes a lot of THC, or greater than 0.3% THC by weight, we would call it marijuana. Whereas if it made less than 3% THC, we would call it hemp. So with the approval by the 2018 Farm Bill of hemp production, we ended up with an excess of plant materials that served as a source of CBD oil. And that hit the market and was unregulated because they determined that if it had less than 0.3% THC, it was not marijuana and would not be regulated. So as a result, 5 or 6 years ago, we had this explosion of CBD oils being sold over the counter and marketed as cures for a variety of disorders. Unregulated over-the-counter sale. Well, these OTC cannabinoids exist in, or over-the-counter cannabinoid products exist in, a regulatory gray area. The FDA does not regulate them, although they say that they cannot be called a food or dietary supplement because they haven't been proven for safety. But there are these regulatory loopholes, and some of them are being exploited to produce products that contain delta 8 THC, a synthetic compound that is also unregulated at this point in time. These gaps leave us with a significant problem where consumers are vulnerable to the marketing of these OTC products, that marketing not being backed up by clinical trial results. And so a number of studies, including our own, have shown that many over-the-counter CBD products are totally mislabeled. They don't contain the amount of CBD that's on the label, and they contain significant amounts of THC, which would make them psychoactive potentially, subject to drug-drug interactions, and in fact, a failed drug test because you don't know what's in the product because it's not regulated the way prescription drugs are. More importantly, those same over-the-counter compounds can contain contaminants, again, because they're unregulated. And so here's a study in which the investigators took a variety of different over-the-counter products, each represented by a single point on these curves or on these graphs, and showed that there were heavy metal contaminants in some of them, residual solvents from their extraction, organic solvents, and even pesticides from the plants, from the hemp plants from which they were derived. That's not going to be true of all products, but because it's not regulated, it creates this gray area and a source of potential harm. And that's because prescription drugs are carefully monitored for microbial testing, the presence of contaminants, their potency, as well as a shelf life of the product that's found in them. And all of this comes from the fact that the FDA requires of prescription drugs a variety of heavy constraints on how something is grown, how it is studied in the laboratory, how it is used in the clinic, and how it is manufactured and distributed. None of this is required of over-the-counter products. And frankly, there are unscrupulous entrepreneurs out there that are exploiting this excess of CBD oil, for instance, that's on the market because of the Farm Bill of 2018. So our take-home message today is that there are many different types of cannabinoids, both from inside our bodies, from plants, and from chemical synthesis that interact with our endogenous cannabinoid system, or ECS. And they can influence physiological process and exploited processes and be exploited for therapeutic effects. This long persistence of cannabinoids, because they are so lipophilic, so aversive of water, they hide out in our adipose tissues, produces variable effects across individuals and potential drug-drug interactions because they're metabolized by the same enzymes that many of our prescription drugs are. So we must be careful of that and cognizant of it. There is a fundamental gap in the evidence standards between prescription grade and over-the-counter grade cannabinoid products that we must inform our patient population of. And finally, the prescription grade and over-the-counter cannabinoid products are held to very different manufacturing, testing, and labeling requirements, and we must be careful about their use, especially particularly in my case, I'm worried about elderly populations where there may be 8, 9, 12 different prescription drugs being taken by that patient, and the addition of a cannabinoid product without knowing what's in it can have severe side effects. So thank you very much for your time. Delighted to have spent some with you.
Thank you for listening. Remember to download the slides and